Hi, I'm Elsa Flores. I'm the Associate Center Director for Basic Science at Moffitt Cancer Center, and I'm here at SITC in Houston. And today I'm going to tell you about my poster, which is Engineering Cellular Backpacks to Deliver Therapies to Undruggable Targets like P53. My lab has been interested in understanding the P53 family. And one of the very interesting things is that P53 is part of a family of genes including P63. Now, delta N P63 is highly amplified, it's oncogenic and found in many cancers. So what we've done is, using genetically engineered mouse models, we've knocked down delta N P63, and you can see that there are fewer tumors. Now, a number of years ago, we were interested in identifying compounds that could be used to knock down delta N P63. And we identified a drug known as pramlintide, which is used in type 1 diabetes. Now, when we treated mice with pramlintide, what we found, now these mice have UV-induced squamous cell carcinoma. You can see that pramlintide reduces the tumor burden of these mice, but not completely. There's some resistance. We also found that pramlintide is effective in small cell lung cancer. So you can see that after treatment here, the tumors are reduced. But again, it's not effective in all tumor types. So then we did a whole genome CRISPR screen to figure out, are there other drugs that can be used in combination with pramlintide? And of course, we found a number of pathways, including the P53 pathway, other cell cycle pathways, and so we started combining pramlintide with other drugs, and that's where we are with that study. Now, we're also interested in using these cellular backpacks, and so these are nanoparticles that are shaped like discs that are placed on immune cells that can carry in drug treatments. And so we've been trying to identify immune cells that can be used to carry these backpacks. Now, we found that MDSCs, which are important for T cell function, can be inhibited by pramlintide in some situations, and then are not effective in others. So we've been tracking these MDSCs in mice, and we find that they go to the tumor site. So these are mice that have tumors on their flank, and also go to the lung. So these are an ideal immune cell that can be used as backpacks. So we're collaborating with one of our bioengineering colleagues at Moffitt, Michael Dunn, who's been a pioneer in this cellular backpack space. And we're using his technology to deliver these drugs to the sites of tumors shown here. And this is really done to reduce toxicity. So right now we're in the preclinical stages, treating different mouse models, and hopefully next year we can share some of our new results.